For centuries, people have wondered if changing what we eat could help control cancer. The idea is simple. If cancer grows by feeding on our body's resources, maybe we can slow it down by cutting off its fuel. This belief has inspired countless diets and remedies, all hoping to find a dietary cure for cancer. The appeal is obvious. Fighting cancer with food feels empowering and less harsh than chemotherapy or radiation. But as science has advanced, we've learned that cancer is not an outside invader, it's our own cells, expertly hijacking our body's systems. These rogue cells are survival experts, making it nearly impossible to starve them without harming healthy tissues. The challenge isn't just about denying cancer food, it's about the complex reality of our own biology. Cancer cells adapt and thrive even when resources are scarce. The dream of simply starving cancer is far more complicated than it first appears. Our quest for a dietary solution continues, but the answers are proving elusive. In the 1920s, Dr. Otto Warburg made a discovery that changed cancer research forever. He found that cancer cells consume glucose at a much higher rate than normal cells, even when oxygen is plentiful. Instead of burning glucose efficiently, cancer cells use fermentation, producing lactic acid, a phenomenon now called the Warburg Effect. This means cancer cells are addicted to sugar, burning through it rapidly to fuel their growth. Warburg's insight suggested that cancer's metabolism could be its weakness. This discovery still shapes cancer research today. PET scans, for example, use radioactive glucose to highlight tumors, since cancer cells soak up sugar faster than healthy cells. Every PET scan is a direct application of Warburg's work. The Warburg Effect inspired decades of research into starving cancer by cutting off its sugar supply. But could this metabolic difference be exploited for treatment? Warburg's findings laid the groundwork for a new way of thinking about cancer. The question became, can we use cancer's appetite against it? The Warburg Effect led many to believe that cutting sugar from the diet could starve cancer. Diets like keto, Budwig, and alkaline became popular, each promising to deprive tumors of fuel. But rigorous studies showed these diets don't shrink tumors or cure cancer. The body tightly regulates blood sugar, making glucose even if you eat none. Tumors still get the fuel they need no matter what you eat. The body's own systems outsmart dietary restrictions. Simply changing your diet isn't enough to outwit cancer's resourcefulness. Cancer cells are ruthless competitors for nutrients. They outcompete healthy cells for glucose, even when food is scarce. This is why cancer patients often lose weight and muscle, a condition called cachexia. Tumors hijack the body's metabolism, hoarding resources for themselves. Restricting calories harms healthy tissues more than the tumor. Cancer cells increase glucose transporters, making them even better at grabbing sugar. Starving the whole body to target cancer is like cutting power to an entire building to stop one noisy neighbor. The real challenge is to selectively block the tumor's access to fuel. We need a smarter, targeted approach. Recent discoveries about brown fat may offer a new way forward. Brown fat, once thought to disappear in adults, is actually present and active. Unlike white fat, which stores energy, brown fat burns it to generate heat. Its secret weapon is a protein called UCP1, which turns calories into warmth instead of storing them. Cold exposure activates brown fat, making it a powerful glucose and fat consumer. PET scans revealed brown fat in adults, sparking new research. Brown fat acts as a natural calorie-burning machine, removing sugar and fat from the blood. Scientists wondered, could brown fat outcompete tumors for glucose? This question opened a new chapter in cancer research. Researchers tested whether activating brown fat could starve tumors. In mice, keeping them cool switched on brown fat, which then consumed large amounts of glucose. Tumors in these mice grew much slower than in those kept warm. Brown fat didn't attack the cancer directly, it simply outate it. This was a new way to fight cancer, not by poisoning it but by depriving it of food. Human studies showed that cold exposure activates brown fat, which soaks up glucose. Early evidence suggested tumors might get less fuel when brown fat is active. The idea of using the body's own tissues to compete with cancer was gaining traction. Could we make this effect even stronger? Activating brown fat with cold or drugs has limits. It's not practical for patients. Scientists turned to engineering. What if we could create superfat cells that constantly burn glucose? Using CRISPR, they modified beige fat cells to keep their calorie-burning machinery permanently switched on. These engineered cells, called ENG beige, act as relentless glucose sinks. The vision? Implant these cells near tumors to starve them of sugar. This approach is part of a new wave of cell therapies, using living cells as medicine. Instead of attacking tumors directly, these cells would outcompete them for fuel. Researchers implanted ENG beige cells in mice with aggressive colorectal cancer. The engineered cells were placed in special capsules near the tumors. These cells immediately began pulling glucose from the bloodstream. Tumor growth slowed dramatically, almost stopping altogether. When mice were switched to a ketogenic diet, tumors used ketones instead, and the therapy lost its effect. This proved the cells worked by starving tumors of glucose. The experiment showed it's possible to use engineered cells to outcompete cancer for nutrients. The therapy was localized, avoiding the side effects of whole-body starvation. This breakthrough points to a future where we fight cancer with metabolic traps, not just drugs. The success in animals is a major step forward. Could this strategy work in humans? Turning this discovery into a real treatment for people is a huge challenge. Safety trials must ensure the engineered cells stay put and don't cause harm. Scaling up the process to make it affordable and practical is another hurdle. The therapy may work best for cancers that rely heavily on glucose. Still, this research combines the best of modern science: metabolism, gene editing, and cell therapy. It offers hope for gentler, more targeted cancer treatments. Each step, from Warburg's discovery to today's engineered cells, brings us closer to new solutions. By exploiting cancer's unique weaknesses, we may finally outsmart this disease.